Hello, everybody, and I want to start by thanking the organizers, so the London Stem Cell Network and also Takara for organizing this workshop. And as Rob mentioned already, most of the work, or actually all of the work that I'm going to talk about today was done as part of my PhD and then a short postdoc with Robert Semple. So really all the credit for supervision, etc., goes to him. And in the spirit of this workshop, most of my slides will be about the gene editing process and tips and tricks, but I will spend a few minutes at the start just giving you a bit of a flavor of the type of work that I'm doing and why I chose iPSCs as the relevant model system for my work. So I'm a PI3 kinase signaling person, and you may be aware that the PI3 kinase signaling pathway is essential for the survival of both human pluripotent stem cells and mouse pluripotent stem cells. And quite nicely, a few years ago, actually fairly recently, it was shown that the PI3 kinase pathway is essential for survival of iPSCs by means of a CRISPR screening haploid human pluripotent stem cells. So if you knock out components of the pathway that lead to its activation, you get decreased survival in the stem cells. And if you knock out components that are negative regulators of the pathway, you get increased survival. This is a very oversimplified schematic of the pathway, but really for the purpose of this talk, what you need to remember is PIK3CA, which is the component I'm working on. And this is the gene encoding the catalytic P110 alpha subunit of the PI3 kinase alpha enzyme. And most people in the audience are probably familiar with AKT and mTORC1, which are common effectors that are studied downstream of the PI3 kinase enzyme, but it's important to stress that there are many other effectors downstream in this pathway. So the reason I got into studying PI3 kinase signaling is my PhD supervisor's co-discovery of a rare, non-cancerous overgrowth disorder caused by mosaic activating mutations in PIK3CA. So the lady you're seeing here has got a massive overgrowth of her lower body because she's got a heterozygous activating PIK3CA mutation, which is not present in her upper body. And following on from this discovery, it quickly became clear that such mosaic activating PIK3CA mutations are the cause of a spectrum of related developmental disorders, which we now refer to as PROS, for PIK3CA related overgrowth spectrum. It's important to emphasize that this is a benign disorder and yet highly debilitating. As you can see, it starts to affect children as soon as they're born. It's acquired embryonically. So as I mentioned, it's developmental and treatments are currently not available apart from surgery. So there is a critically unmet need for developing more reliable and new model systems for preclinical studies in relation to PROS. And that really brings me back to the aim of my PhD, which was to determine the cellular consequences of activating PIK3CA mutations in a developmental context, which is the reason why I chose pluripotent stem cells. The way I went about this was to use CRISPR-Cas9 to knock in an activating PIK3CA mutation, H1047R, the most common and strongly activating one, into the endogenous PIK3CA locus. And originally, the aim was really to get this knocked into one allele, so heterozygously, as would be observed in PROS. But serendipitously, and quite luckily, actually, I managed to get lots of homozygous knock-ins, and that proved crucial for the discoveries that we then ended up reporting. Now, quite nicely, when you have heterozygous versus homozygous H1047R knock-ins, you manage to get a graded activation of the PI3 kinase pathway, which allowed us to study dose-dependent signaling effects in pluripotent stem cells. I won't go into the details of what we actually found out because I want to spend more time on the gene editing procedure, but just highlight how important it is to be aware of allele dose-dependent phenotypic effects of any mutation of interest, really. I think what we discovered, the fact that homozygous but not heterozygous PIK3CA H1047R can lead to a near binary phenotypic switch where you get self-sustained stemness. That's just one single copy extra from the heterozygous state. It really goes to show that there are very subtle quantitative differences in signaling pathways that can be interpreted into and translated into vastly different phenotypes, which is very important when it comes to interpreting disease-relevant phenotypes. When it comes to the gene editing procedure and generally working with iPSCs, when I was preparing this talk, I was thinking a little bit back to when I had to do this, which was now 6 years ago, so in 2015, and what advice I would have given to myself. And I jotted down a few considerations, generic ones and also specific to knocking in point mutations using homology-directed repair template. I won't go through them point by point because the presentation will be available afterwards, but I will highlight a few key bits of my protocol and especially emphasize what has been said already, the importance of really using a well-characterized iPSC line, including checking its karyotype and doing all sorts of initial checks such as its single-cell survival, tolerability, dissociation reagents tolerability, nucleofection protocols, etc. So a bit at the risk of sounding, well, weird in a sense, I've put this figure here to sort of illustrate that it's very important to start off with a solid base in order to get reliable results with iPSCs. Those of you already working with them will know that it can be quite a noisy system. So having these systematic optimizations in place is very important. I'll skip this in the interest of time. It's basically to demonstrate that I was practicing what I'm currently preaching, so testing optimal medium conditions, coating reagents, clonal density seeding, etc., prior to starting the gene editing protocol. This is the overview of my knock-in strategy. It's relatively simple. It's a single nucleotide change in the C terminus or 3' end of the transcript and gene. I used 2 templates, and I'll come back to why I needed a second template. The first template is the one that actually has the mutation of interest, but it's also got 2 silent mutations. The reason being that you want to prevent Cas9 and the guide RNA from cutting your template, but also from recutting a successfully targeted allele. Now, the previous speaker also touched a bit upon expansion of multiple clones, but this really cannot be emphasized enough. It's really important to have multiple independent clones, and especially to have matched wild-type controls that have gone through the exact same gene editing procedure. Forcing these cells through a so-called bottleneck selection in the process of single-cell cloning can really result in a barren phenotype. So it's crucial for your downstream phenotyping. Otherwise, you may be comparing apples to pears, as it were. A bit of potentially useful resource, the stem cell reports paper is a good guide if you want to know more about tips and tricks for experimental design and interpretation of iPSC-based disease modeling, especially whether or not you need multiple different iPSC genotypes, which will partly depend on the research question. For the gene editing protocol, I've provided a step-by-step approach in previous block series for benchling, but actually most of the things that I'm going to cover in the next slides are outlined in much more detail in my PhD thesis, which is free to download and look at. So I would probably go to that if I have further questions after these slides. I'm quoting my protocols as being old because this is obviously back from 2015 and 16, but actually most of the things that I did back then is also exactly how I would be doing it now, and I'll highlight where there may be potential differences to make the process even more efficient. So this is an overview of my approach. And before going into the details, I just want to clarify a few technical terms. So when I mention the target strand, that's basically the strand that is complementary to the guide RNA, and obviously the opposite is the non-target strand. This is an important point because when the wild-type Cas9 cuts, you get an initial release of the non-target strand first and in asymmetric fashion, which is quite important for the way in which you design your homology-directed repair template. So the design obviously splits in 2. Once you've identified your locus of interest, I won't go into every single detail, but obviously be mindful of how far away a single guide RNA cut is from the site of your point mutation. This is critical for efficiency. The way I delivered the single guide RNA sequence into cells was by using a so-called G block. These can be ordered commercially and are fairly cheap. It's basically a block of DNA that encodes the guide RNA with some additional modifications that make it more stable and better expressed in cells. And that was actually quite important when I was initially testing everything in a simple cell system such as hexahelix. This approach was far more efficient than the conventional or the old approach, which is to have the Cas9 and the guide RNA encoded in one plasmid. Nowadays, this is one modification that one could introduce, is to just use RNPs instead. And this is the more costly option, but I'm currently using it. And I would also recommend actually using the complete single guide RNA, a synthetic single guide RNA, so that will even avoid the need for pre-complexing that you would otherwise have to do if you order a two-component system. As the previous speaker already touched upon, I would recommend testing multiple single guide RNAs if you're working with a completely new system, and obviously also to test it in prior to going into IPSCs for checking whether or not you have got sufficient cutting efficiency. For the HGR template design, for point mutation, you basically only need a single-stranded ODN, so it's fairly easy. You can go for the rational design approach, which is what I briefly alluded to earlier, where you can design an asymmetric HDR template that's identical to the target strand and will hybridize to your non-target strand. And the reason it's asymmetric is because of this release mechanism. And there are more details in this reference, but also, as I mentioned, in my own work. I've touched upon the silent mutations already, and they're important, so I won't go into further details of that. But moving on to how you can control the zygosity, it's actually rather simple, and that's why I needed a second repair template. Because if you mix your template containing your mutation with a template that only has the silent mutations at a 1-to-1 ratio, you can increase your chances of getting heterozygous mutants. Otherwise, given the efficiency of the Cas9 when it actually enters the cells and cuts, you're more likely to just get biallelic mutations, except this may also, or this can depend on how close to your target site the single guide RNA cut is. In my case, it was very close, so I pretty much got only biallelic homozygous knock-ins if I only use this template, but you can play around with the distance to then favor homozygosity versus heterozygosity or vice versa. And this is a useful article for that. Especially because IPSCs are so expensive to culture, it's very important from the start to have a think about how you're going to screen your clones in an efficient manner, how many you need to expand in order to have a decent final number, and aim to have a much higher final number than what you think might be necessary, because there is a high attrition rate throughout this process. IPSCs are generally very fragile, and I found that mine were not very happy going through faxing, so I went for a CYP selection protocol for enrichment of my mutants. It's based on a methods paper from Bruce Conklin's lab in 2014, which I then modified to suit my needs. And again, it requires testing in your cell line of interest to determine the minimal number of IPSCs that you should seed in a 96 well in order to get at least one cell surviving. The way I did it was to nucleophect myself and plate them in a 96-well plate screen, which I'll come back to in a second, identify the wells that were positive from the mutation at the bulk population level, then subclone those individual wells into quite a large number of 96-well plates, and then go through this process again for subsequent screening and expansion. Usually, if the subcloning process has worked efficiently or well, you should be able to just go straight to the expansion without having to do a second step of subcloning. Now, to the screening, again, if you can, try and introduce a restriction enzyme site when you're knocking in your mutation of interest. Then you can do a so-called RFOP assay, so restriction fragment length polymorphism assay. I was lucky because my mutation actually introduces it, but I also made sure that my silent mutation would introduce yet another restriction enzyme site. So the way this protocol works is that you can then amplify your locus of interest with two primers, one that is FAM labeled, and if you've got the mutation in your population, then you can incubate your amplicons with the relevant restriction enzyme, and they will then cut those that have got the mutation. Well, the enzyme will cut those amplicons that have got the mutation, and you'll be able to separate them on a gel, if the process has been efficient, that is, because otherwise you won't be able to pick it on a gel, but you still probably will be able to pick it up by more sensitive nucleic acid analysis methods down to 1% knock-ins. You should be able to pick up with this. Now, this is more efficient or quicker than conventional, Sanger sequencing, unless if you're the Sanger center and you can do large-scale sequencing. at little cost. But this can be made even more efficient, and that's something I worked towards at the end of my stay in Rob's lab, where I was aiming to optimize or set up a co-selection method relying on the knock-in of a luminescent peptide tag onto an extracellular surface protein on the iPSCs, which would then allow me detection of successful knock-ins by simply screening for luminescence. And this, importantly, can be done in live cells and takes less than 30 minutes, so you don't have to replica plate or to extract DNA from the cells. The thinking behind this is that if you have got successful knock-in of the hybrid tag and you simultaneously introduced reagents to target your gene of interest, then it's more likely that that would also have been targeted if the hybrid has gone in successfully. So again, this is the reference for this method, which is really just at an early stage and is provided for others to build upon it. Finally, when it comes to QC steps, one thing is getting your clones or confirmed that they're a correct genotype. But after that stage, there are lots of other QC steps that, in my opinion, should be done to be absolutely sure that you're working with the right cells. So obviously, karyotyping, they're checking for off-target effects. I would also strongly recommend checking your locus with a matrix of primers that span 2 kilobases or even more around your edit site to rule out larger deletions, which have been reported with CRISPR-Cas9. Those can give you the false impression of getting biallelic knock-ins, but in fact, you could have homozygous mutants instead. I'll skip this point and end by emphasizing the importance of also checking the status of your p53 locus. It has been demonstrated that iPSCs and human embryonic stem cells as well are very prone to enriching for a loss of function p53 mutations in culture. And in fact, we also found some in our wild type, so-called PIK3CA wild type clones, once they had gone through this entire bottleneck selection. So again, these could be hidden confounding factors if not checked for. As a signaling person, I think I will end by also emphasizing that one way in which you can actually increase the signal to noise ratio in your phenotype, downstream phenotyping, is to actually be really consistent with your cell culture and also timing of your experiments. This is just an example to show you how one of the key components of the PI3 kinase pathways, AKT, and its phosphorylation changes just as a function of time. So these are my homozygous mutants and these are the wild types. And you can clearly see, in response to different inhibitors as well, that the ratio, the difference that you can measure between the mutants and the wild types will differ substantially based on timing. So when you're doing experimental replicates, it's important to pay attention to this because it could lead to more noise than you desire. And the same applies at the transcriptional level as well. This is partly due to changes in nutrient levels as a function of culture time, but also confluency. So I think I'll end here by thanking my PhD supervisor, Rob Sempo, and a very talented research assistant in his lab who was helping me with all the iPSC work, and obviously the funders, mainly Wellcome Trust.